A replication is very intricate process, and there are many chances of a mistake. Sometimes some wrong nucleotides are incorporated in the new strand. But the replication machinery achieves a remarkably high degree of accuracy using a proofreading mechanism. It has its own proofreading mechanism which removes wrongly incorporated nucleotides. However, this proofreading is not foolproof. Some misincorporated nucleotides escape detection and become a mismatch between the newly synthesized strand and the template strand. So there may occur some mismatches. If the misincorporated nucleotide is not subsequently detected and replaced, the sequence change will become permanent in the genome, which is called mutation. During a second round of replication, so in the first round, a misincorporation has taken place. Base has been incorporated wrongly, but in the second round of replication, the misincorporated nucleotide will direct the incorporation of its own complementary nucleotide into the newly synthesized strand. At this point, the mismatch will no longer exist. Instead, it will have resulted in a permanent change, a mutation in the DNA sequence. So in this way, in the first round of replication, a nucleotide is maybe mismatched, but in the second round, that mismatch results into a permanent change in the sequence, so which is called a mutation. So here I can try to explain you a little more about this change, about this misincorporation. This is a double-stranded DNA. It has some sequence, for example, A, T, G, C, C, T. On the other strand, it will have A, G, G, C, A, and T. So during replication, the strands are open and new strands are synthesized. So for example, we say here is T and then C, C, G, T, A. Here in the new strand, in front of T will come A, but maybe in front of C, G should come, but due to incorporation here, it may come T. So after replication, two new molecules will be formed, and these two molecules will have the sequence as A, T, G, C, C, T. This is parental strand, and in this strand there is misincorporation, and in front of T is A, and in front of C here becomes T incorporated, which is actually misincorporation. The rest is the correct. So G, C, A, and T. So in the next replication, this strand is same, A, T, G, C, C, T, and the other has a misincorporated nucleotide, which is A, T, G, C, T, A. So when the second round will take place, this strand in the second round, the complementary strand of this strand will be T A G C A T. So in this strand, here becomes the permanent change. So this is a mutation. So up till here, it was misincorporation, but in the second round it has become a mutation. So in this way, these misincorporations lead to mutations.